Well, welcome everyone to Keola. I'm sure you've had a lot of this before. We're really pleased to host this workshop through the Centre for Biodiversity Analysis. We run training workshops periodically depending on interest and demand, and clearly there's a lot of demand for this one. So my personal thanks to Lewis and Bernd and the whole team who did a lot of the heavy lifting to put this together. So I'm introducing the morning session on lineage divergence / phylogenetics, focusing on SNP data. And I actually don't know how to do anything, so I'm doing a general introduction to some of the concepts and some of the gnarly issues that we face in doing this sort of research. But the people who do know how to do stuff are Indiana Jones, Sally Potter, there's a future fellow at Macquarie, and Dumbledore over there, who you all know, Arthur. So they'll be following up my initial conceptual comments with practical details and more worked examples. There's an online tutorial that they've developed that goes along with this. Sally's end piece in particular will lead into that tutorial, right? Or cover some of that ground. All right, so I'm very happy to take questions as I go along. As I tell my students, the more questions you ask, the less I cover, the less it's on the exam at the end. You do know there's an exam, or pop quiz. Okay, so this is roughly what we're going to cover. I'm just going to blather on for 15 minutes and outline some issues. Sally will then take over and look at some of the types of phylogenetic analyses that you can do using SNP data. Arthur will focus on the very fundamental question of what are the OTUs? What are the things amongst which you're trying to determine relationships? Not necessarily species, they can be lineages, they can be genetic populations. Arthur will cover some of that. Sally will then come back and take you through in more detail some of a sample of the methods that are out there in the literature. There's a hell of a lot more. If you just look at the recent issue of Systematic Biology, came out this morning, there's another whole set of methods in that. So it's a very rapidly developing field with some very smart mathematicians underpinning it. And then we'll come back, I'm down to have discussion at the end. I actually don't have much to say, so basically I want you to think about the questions and issues that have arisen when you think about the systems you're studying and try to fit it into one of these boxes, and we'll just have a general discussion at the end. Okay, so I think Sally's got this slide as well. This is a terrific review paper focused on SNPs. There was a burst of energy, much of which Adam Lachey led. Adam actually participated in one of these workshops a few years ago. For us, hosted at University of Canberra. And he, with Jamie Oakes, put together a really nice summary paper, just as SNPs were really emerging as a really practical data type to be using in population genetics and phylogenetics. And he unpacked some of the opportunities and some of the issues around using SNPs in phylogenetics. So you're all familiar with the SNP matrix. Note here, all these loci are variable. It does not show the invariant sites. And I'll come back to that later. Those of you used to getting just data provided to you by Dart will only have the invariant sites or the tags that contain invariant sites. So I want you to think about that. And then, again, this is just a small sample of methods that Sally will introduce you to. Concatenation, there's a whole bunch of likelihood-based methods that are very practical, they're quick. They tie into models of molecular evolution in really interesting ways. That's a very mature field. Rob Lanfear, who's here somewhere, probably off getting coffee. Oh no, you caffeinated, Rob? Oh, good, excellent. Just looking after my colleague there. Rob Lanfear has led a lot of work here in defining models of molecular evolution and selecting models of molecular evolution for phylogenetics and other things beside. And these two are different in flavour and concept. These are species tree analyses. So here, the tips can be individuals, they can be species. Here, we're grouping individuals into what we call species, and Arthur will come back to what do we mean by a species. And we're inferring the relationships among the species given the gene trees or site patterns within the data. So a couple of different methods to do that. One laborious and slow, but full Bayesian, that gives you all sorts of parameters like population size, divergence times, posterior probabilities. And SVT quartets just basically gives you the topology. Although there's some things you can do beyond that. All right, so I'd really encourage you to go back and read that paper. Did we provide that as reading, Sal? No. If not, you can look it up. It's in annual review. Yeah, good. It's a really good starting point to just get into it. So just a couple of conceptual issues and thinking about what's the domain we're in here. So you've had lots of population genetics, you've had landscape genetics, which is all here in the upper left of the diagram. And this is pretty obvious, right? This tends to occur over fairly small spatial scales and fairly small temporal scales. That is time, the amount of time elapsed. So often you're interested in contemporary processes of population size, adaptation, and structure across a domain. As we go into phylogeography, John Levis and colleagues call that the sort of bridge between pop gen and philo. And one of the things that I want to come back to right at the end of this workshop is the merging of population genetics and phylogenetics that's enabled both by the data, but also the underpinning theory, particularly coalescent theory that Sally will introduce. So phylogeography is really intraspecific lineages primarily, and phylogenetics, of course, goes from lineages, species, up to whole clades, macroevolution type questions. And I've always thought that the space for snip-based analyses is phylogeography, or pop-gen phylogeography, to sort of recent phylogenetics. We don't look at amniote relationships or archaeal versus bacterial evolution using SNPs. We use whole genomes or something else for that. But I'll come back to that in a minute, and the reason why. So phylogenetics is about the relationships between independently evolving lineages. Trying to figure out their divergence times, the sequence of divergences that yield a phylogenetic tree. But the reality is something more like this, of population splitting, merging, some level of gene flow, particularly early in the divergence history, and potentially gene flow between non-sister taxa or more distantly related species occurring at some period in evolution. And I think it's fair to say that as we've applied SNP data and genome sequence data now to many radiations, we're realizing that interspecific gene flow, even between more distant related things, is actually relatively common in the history of life. And it's a living nightmare for phylogenetics, because a lot of our models are based on the idea of bifurcating evolution, not reticulation. So for those of us, particularly who are interested in recent speciation processes, this is a substantial complication. The question is, how much does it matter? So I want to illustrate my own little journey on this through a case study, which sort of shows you some of the complexity, but also the opportunity. And it's sort of born of the idea of really extensive sampling. If you're interested in the divergence history of a set of named species, standard practice in systematics for decades has been sample well within that species. So do thorough sampling below, one step below your level of inference. And this can be particularly important in recent radiations, morphologically conservative groups, where often you've got cryptic species that play merry hell with the phylogenetic analysis if you mix them inadvertently. So understanding what's happening from the population scale through the phylogeography to lead into the phylogenetics, I think is very important in terms of the tree you get for these closely related species groups. So sampling below the level of inference is one sort of take-home message, whatever that is. So this is an example. Sally led much of this work, along with my other student, Jess Fenker. And these are little skinky things across Northern Australia. We started the concept that they were a single taxonomic species. Jess did some landscape genetics, but you couldn't do that across lineages, so she applied structure analyses and PCAs and all that stuff to find what were the genetic clusters within which you could sensibly do landscape genetics. Because landscape genetics has no concept of divergent evolution. Right? It's in well-mixed populations. So then, this feeds into a comparative analysis across a clade of these skinks. There's 1, 2, 3, 4 nominal taxonomic species here. When Sally looked at these more closely with exon capture, this is SNP data, this is exon capture. You can see that there's, it's not a random mating group, there are multiple lineages within them. We applied various lineage delimitation methods, and yes, in Carly AMAX, there's 4 things that are quite discrete, and they become our units for phylogenetic analysis. They're sufficiently independently evolving that we think, yeah, okay, we'll run with that. And then Jason Bragg and Hugh and so forth then generated a phylogenetic tree for this entire family in Australia, including the species, but also the intraspecific lineages within those species. I should point out this was a fully Bayesian, star-beast analysis with 2 sets of 100 loci. It took about a year to run on the computer. So it's not for the faint of heart. Don't just. But actually, most of the results were obvious in a few-hour concatenation analysis. Just saying. Okay. So I've mentioned, I'm sort of hammering on this point of reticulation because it's a complete model violation for some of the species tree coalescent-based methods. And it can also mess with concatenation studies. So you can have a set of diverging populations. Some might collapse entirely. You get occasional gene flow between lineages at different points in time. You might get hybridization between taxa where they're in contact. And that might be a true picture of a history, and we try and shove that into a box that assumes independent evolution of those lineages. And the question is, how much does that matter? How wrong do we get it if we do that? So figuring out the pieces and how they've diverged is really very important. So this is another example, again, Northern Australia, another little brown lizard that no one gives a shit about, but I love it. Little brown geckos, again, sampled across the tropics. And we started out this work with a taxonomy saying that all these things up here and all these things down at the bottom left were a single species, and that turned out clearly to be wrong. There were lots of other species morphologically distinct species nested within that group. And these ones were pretty simple to delineate. They were overlapping geographically with other taxa, no gene flow, with SNP and exon capture data. Post hoc, we found morphological differences that made them diagnosable. We described them as new species. Easy. This group down here is sort of a species complex. It's sort of left. If you looked at and sort of scratched our heads and said, oh, that's hard. But actually, it's the hard stuff that's fun. If you're interested in divergence histories and speciation processes, it's the messy stuff where the gold is. But it's really difficult to pick it apart, right? And it takes a lot of sampling. So this is one example of an early result. This is just a structure plot between some of these clades where the colours are all screwed up. These two things, the green and the red, sorry for colourblind blokes, merge entirely, so it actually doesn't matter if you're colourblind. They're the same thing. That's easy. These two things here have a lot of integration. They're actually green and red here. Sorry again. And these two things here, we see almost no genetic exchange. So I thought, okay, is it one species? Is it 3 species? Is it 2 species? And how are they related? And I should say this shows a nice, neat phylogenetic tree. You'll notice there's no confidence limits. The relationships in this group here are completely inconsistent depending on how you analyze the data. Concatenation, species tree methods, between different species tree methods. Something was clearly wrong. Very short internodes at the base. Very hard to resolve the relationships. For some things, that doesn't matter, right? You've got a depiction of the evolutionary history. It's clear that these things have separated from each other in over short time intervals, which is exactly when you expect it to be a hard problem in phylogenetics. Sally will talk about incomplete lineage sorting. That's when the gene trees mostly disagree with the actual divergence history, because they don't join up right at the point that two species separate. They join up somewhere back in the common ancestor. But it gets even worse if they're exchanging genes as they go along. So, and indeed that is the case in this group. So this brings in another player. This is the same complex, but now with the sister group. This is a described species, multiporosa, occur in the northwest Kimberley. We found that all the population of Gahaira Nana in here with the open circles had mitochondrial DNA from this guy. Right, so often strong mitonuclear discordance. So you make a gene phylogeny from mitochondrial DNA, that's really easy. You make a phylogeny of whatever sort from your nuclear data. If there's strong discordance, that's often sets up the hypothesis of some sort of integration back in the history. Maybe driven by selection, maybe a purely demographic effect. So we explore that, and this is one method called BPP from Ziheng Yang, where you can specify a phylogeny and test different models of integration or no integration given a divergence history. And it's very rigorous, and this particular model did infer, in fact, that there had been gene flow, and time is on the y-axis here. So somewhere way back towards the initial separation of this group from these two taxa, there was substantial gene flow from multiporosis. And this is in the nuclear data now. Mitochondrial DNA is not part of this. So that's supported the idea there had been a reticulation. And perhaps that in part is why sorting out the relationships among these groups has been so very difficult. Is that you're violating the assumptions of the species tree models. Okay. And then we followed that up now with more detailed analysis of gene flow at geographic boundaries between all these different taxa. So, coming and sampling more intensively here, 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 to see, is there integration or gene sharing when these things are parapatric, or even sympatric in these cases. And in many cases there is, but it's geographically very localized. So are they good species or not? Well, it depends what you want a species to be. There's often no clear answer when you're in the sort of grey zone of speciation. So these two are exchanging genes, clearly the structure plots, exchanging genes over about 10, 20 kilometres out of a distribution that spans about 1,000 kilometres. So they're functionally independent groups, but with a bit of leakage at the boundary. These ones, they're even smaller, just up Mitchell Plateau, we found a few hybrids. These two overlap with no gene flow at all, shown here. So these two, under a biological species concept, are clearly separate species, but there's a bit of leakage between the others. And this is kind of what you expect, right? If you're involved in reproductive isolation, it isn't just clock-like. It's under selection. So some lineages will evolve reproductive isolation faster than others, and you'll get these sort of messy scenarios where, oh, these look like good species, and maybe these ones aren't, but these two sure are. So that's called a paraphyletic species, and that's totally fine with me. Some people don't agree, right? And here I'll be saying, oh, bullshit, Craig, what are you talking about? Let the organisms tell you the story, right? We are imposing statistical methods on a true evolutionary history, which is fascinating. I only want to tease it apart. At least I do. All right, so that was just to introduce some of the complexities you can hit in the real world. And as I say, I love that as an evolutionary biologist, because that's where the gold is. Now, let's turn over to what's the effect on, in simulations of integration on your gene trees and on your branch lengths. So this is a really nice paper. Colby Long and Laura Kabatko. Kabatko is, again, one of the key developers, a lot of these methods. She does fantastic work. And so basically they set up simulations with a divergence history. Here's an outgroup, 3 taxa A, B, C, and they're modelling, it's called an isolation with migration model. So 2 lineages splitting with a continuous rate of gene flow between them, measured by M, which is actually the number of migrants per generation, and asking how often do you get the phylogeny wrong? For example, in particular, putting B together with C to the exclusion of A, right? So the wrong species tree. And of course, it depends on the amount of migration. At some point, migration will just overwhelm it, so that, in effect, biologically, if you've got a very high migration rate for long enough, B and C are each other's closest relatives genomically, but that doesn't reflect their history of divergence. So it again depends what you care about. So these are the results using SVD, which Sally will introduce. Red is bad, green and yellow is not so bad. And these are the migration parameters. So up to very high rates of genetic exchange over that whole divergence history. You get it wrong, pretty much random actually, in terms of the phylogeny you get. But for lower migration rates, just occasional migrants exchanging genes, they do pretty well for a modest number of sites. So by modest here, this is 200,000 sites. This is probably where a lot of us are, right down on the left-hand side here with 10,000 SNPs or something like that. So with the modest data sets we tend to get from, say, DART, rather than sampling SNPs from whole genomes, you probably do need to be concerned that you're getting the wrong tree if there's even modest migration rates. And this is the same thing for using genes rather than numbers of sites. So, and I should say concatenation here isn't noticeably worse than a lot of the species tree methods. So it's not a, you tell me, Rob, it's not really a violation of the likelihood model, is it, for concatenation? Gene flow, or is it? It is, fine, okay. So it sucks no matter which way you do it. But it's pretty robust. Which one of these is? Oh, shown down here. You can see somewhere up here, concatenation. So performing quite well in terms of not being screwed up by this gene flow. So that's good. And then Adam Lachey did another really nice paper. Again, this is about a decade old now, this work. But it's true, remains true, where he looked at different sorts of migration processes. This IM model between sister taxa. Well, between sister taxa it doesn't really matter. It's just going to affect the branch length, your estimate of divergence time. But what do you really mean by divergence time when there's continuing migration between them? That's sort of a conceptual question. Paraphilic gene flow, this is where it's going on between non-sister taxa and then ancestral gene flow, a bit like the first one I showed you. How am I going for time, Sal? Okay. Just tell me to shut up. Okay, so this is just a depiction which I really like of some of the ways it can go wrong. And here the topology often is not so bad, but the width of these branches here is the estimates of population size. These are so-called pipe diagrams, where the breadth of the branches is number of individuals, genetic individuals. But the branch lengths get all screwed up. And again, that should be obvious. If you're exchanging genes during that divergence history, it will appear, the divergence time will appear less than the true divergence time. So it's sort of a systematic bias there. So it depends on whether you care about topology or branch length, or divergence times. I'll just do this very quickly. There's another whole class of methods and models which are applicable to SNP data, where if you've got just 3 or 4 taxa that you're deeply interested in, you can test different models of divergence, models with gene flow, models without gene flow, models with changes in population size over the history of the splits, bottlenecks, all that sort of stuff. And those parameters like bottlenecks do affect the data or the site patterns that you observe in the data. So I actually like using some of these types of methods. Well, I don't do anything. I encourage my students to use these methods to do model selection. So model selection is terribly important. You want to know if you have enough data to even distinguish between different types of histories, rather than just assuming the single model you get is the best. Do you even have enough data to distinguish between gene flow or not gene flow, population size change or not? And you can do that via model selection. And a lot of these are approximate Bayesian computation models mostly. They can tell you whether you have enough signal in your data to even get into that level of inference. And often you do not. But the model selection tends to be really robust. Parameter estimations are more suspect. But this is an example. This is human data, very extensive, of course, looking at human history. You simulate allele frequency spectra or site frequency spectra jointly between 2 taxa or 3 taxa. I think the current methods that are best for doing this with SNP data specifically is this thing called moments. So I encourage you to take a look at this. A really good manual here for moments, and it does all sorts of stuff: selection, divergence, population size change, migration. But again, do you have enough data to go there? Often they need whole genome data with SNPs sampled out. But if you're going really deep into a group, it's worth exploring. And this is one example. I actually found this paper just yesterday. Recent paper in the Journal of Biogeography with the sort of SNP data sets we're used to getting. 5, 10, 15,000 SNPs recovered after filtering across a set of populations. It's a garter snake in the US. And they've done all the standard stuff. This is sort of admixture type modeling using Tessa, which includes space as well as the genetic covariances. An IQ concatenation tree. You can see it's not a history of clear independent evolution across lots of lineages. It's more like a paraphyletic grade. And then they tested 13 different divergence models based on their understanding of the species and the geography, and found one best fitting model, which is like this, which models then the introgression events among tracks. And they did this using moments, that software I just mentioned, for a modest sized data set like we typically get. So they had enough power to do the model selection. What they don't show here is the confidence limits around these parameter estimates, and I bet they're really big, particularly the time of migration as opposed to the amount of migration. So model selection is a really good way to go if you've got a complex history that you're interested in. So just finally, I want to mention something about sampling SNPs. Do I have 2 minutes, 3 minutes? Yeah. So, and are you going to touch on this as well, Sal? Okay. So, I imagine you're all familiar with allele dropout, right? Have you covered that already in this class, Lewis? Have you covered allele dropout? Ah, okay. So you send your cherished samples, you do the field work, you rate a museum, you send your stuff to DART, they send you back a matrix, and you see not every SNP is scored in every individual. Right? There's lots of gaps in that data. And you can actually use the gaps in the data. Arthur might talk about that. But it often looks like this, and this empirical data from fence lizards, from again in this Adam Lachey review. But for a typical data set, you'll get a high proportion of SNPs recovered fairly uniformly across individuals in the most recently diverged taxa. That will drop down as you go to greater evolutionary time, and further down as you go further down, comparing across the base of that tree. And one cause of that, the dominant cause, is the restriction sites themselves that are used to generate DD-RAD data mutate. Like the restriction enzyme needs a very specific sequence of bases, 4 or 6 or 8, depending on the cutter. And back in the old days, we used to actually use the restriction site gain and loss as our phylogenetic data. That was before PCR, if anyone can imagine that. That's what we did. Adam, you did Alzheimer's. You're a living fossil. So anyway, this allele dropout is a very well understood phenomenon from the decay of homology of restriction sites just through nucleotide substitution. And depending on how complete you want your matrix to be, of course, you shed a lot of SNPs. This is real data from the Scoloporus, starting with 100,000 if you only require, if you keep all the SNPs, if you allow only to have 10 of the 70 individuals, you get about half the number, and so on down. If you require all the individuals to have it, you've got a tiny, tiny data set across a modest phylogenetic depth. So what do? How do you filter the data? And I was actually surprised by this result. I would have thought, I always like to have complete data or nearly complete data, robust and that sort of thing. And this was a lovely study by Huang and Knowles, again around the same time, simulating SNP evolution across phylogenetic trees, and then looking at allele dropout, and as both a consequence of restriction site loss and also post-filtering for coverage, excess heterozygosity, the sort of things you've heard about already in this workshop. So on the left-hand side is a panel just showing a set of loci. We've depicted here across individuals on the y-axis, loci on the x-axis. The number of bars here is the amount of coverage. You process according to minimum coverage. You process according to remove things with excess heterozygosity. They could be paralogs, right? Copies of different genes rather than alleles from the same gene. And you end up with this spotty, patchy data matrix. And this again shows the threshold for missing data, the minimum number of individuals, and the size of the data matrix. So as you require fewer and fewer missing individuals, so this is complete data, this is patchy data on the left, you can see the loss of the reduction size of the data matrix with higher low coverage data. This is for a recent divergence history. N is the effective population size. So this is two lineages that have diverged over 2n generations. On the right-hand side is a much deeper history where the divergence time is 20n generations. And so you've got a lot more loss of restriction sites in the data and also post-filtering loss as well. Does this matter that we're biasing our data towards the more slowly evolving loci? And their conclusion in this study, and of course it's a simulation study, it's one particular set of conditions. It doesn't tell you if there's a general result, but it's an interesting result. That the RF distance here is the difference between your recovered topology and the true topology in the simulation. So a big number is bad. And here, this is the tolerance for missing data. Okay, the very last thing is that's all about topology, this sort of sequence of divergence events. What if you're interested in branch lengths? And we often are for macroevolutionary studies, rates of speciation, all those sorts of questions. And their integration can be really, really bad. Oh, sorry, this is not integration. Missing SNPs can be really bad. So I just said that for topology, you can have a lot of missing data, and in fact, it might even improve. Not filtering that might even improve your phylogeny. But again, this simulation study by Adam Lachey again found that the more you filter out the missing data, the better. Sorry. Missing data? No, this is about invariant sites. Let me back up. Different thing. So the branch lengths here, the question here is, what happens if you just include your SNP matrix versus if you include all the invariant sites? Now, if you get a data matrix from Dart or other AGRF or whoever, you'll sometimes get the Dart tags, you'll get the sequence, and you'll get the SNP positions within that. What you don't get is the bits of the genome that were sequenced but found to have no variation at all. So you've already got a biased data set. You don't have a proper accounting of the invariant sites. And this is something we've talked about a lot with Bernd and Arthur. How do we deal with this for things like heterozygosity estimates? You heard from Renee about that. It does have a profound effect on branch length and phylogenies if you improperly account for the invariant sites. So the best way to get that, of course, is to map your SNPs to a genome. so that you know what sites are, what the invariant sites are from your raw read data. But otherwise, there's various corrections proposed, a couple are applied here, and basically the more missing data you have on the left, the bigger the branch length inflation. And this problem's been known for over 20 years. And really, I think the only way around it is to map reads to a genome and get a proper estimate of the invariant sites. Now, you can go some way towards it with modelling it through a molecular evolution model, and that's what's shown at the top. And at the bottom, they're actually including the invariant sites in the SNP tags. So, but again, red here is really bad. That's 500-fold, five-fold rather, overestimation of branch length by leaving out the invariant sites. So think about that.